So welcome to everybody. This is my name is David Bogle. I'm a former scientific vice president of the European Federation of Chemical Engineers. I am a professor at UCL, and this is the webinar of chemical engineering as applied to medicine, and some of you will know that I was quite instrumental in helping to set this up. So this is this series of webinars has been going for about 4 years. It's in its 4th year since, and we've had a series of webinars over in groups of a dozen or so, once or twice a year. This time, over these 2 weeks, we've got 9 of our working parties and sections have webinars, and we're very pleased that the 2 newest working parties sections, of which this is one, where chemical engineers applied to medicine and early career chemical engineers, are doing a webinar during this series. And it allows people to not only follow their specific interests, but also taste other areas within the European Federation. Our working parties and sections cover pretty much all of chemical engineering. So the links are available on the invitation. So if you want to check into some of the others, you're most welcome. So the working parties and sections are very much the scientific engine of the European Federation of Chemical Engineers. I can't remember how many there are now, 25 of them. I mentioned the 2 new ones. And I might also mention that one of the key events alongside these webinars, one of the very key events is our biannual conference, the European Chemical Engineering Conference on Chemical Engineering, which will be in September in Berlin. So I encourage you all to think about, it's too late to submit a manuscript, but it's not too late to submit a poster, and of course not too late to attend. And it's in mid September in Berlin. So I just will finish now by thanking everybody involved in the working party and sections, in particular in this particular one, Tomasz and David Amanka for setting setting up the program for this webinar, and of course thanking very much to Martine, Scientific Secretary of the European Federation of Chemical Engineers, for all the huge amount of work she's done setting up these conferences. So with that, let me hand you over to the chair of the chemical engineering, the section on chemical engineering as applied to medicine, Tomasz Sosnowski.
Okay, thank you, David. Good afternoon, everybody. I'm very happy that you attended this webinar, which we start in a few minutes. We start the first presentation, but let me say a little bit about the section, how it was created. And this is I want to thank David, who was very enthusiastic about this, about creating this section. I think that this is very important, that we can use our chemical engineering skill set to do something not only for the industry but also for human health. So this is what is this section for. And we had the kickoff meeting in December last year in Paris, when we had the opportunity to listen some presentations. But this webinar is a continuation of this activity. And I believe, as David mentioned before, that during the conference, which will be the next conference in Berlin, we will have more opportunities to see that this topic is really within the chemical engineering. And before I pass the microphone to our first presenter, let me only show the screen about the section. I prepared it just to show this. The name, the chemical engineering as applied to medicine. This is the official name of the section. The abbreviation is ChemiMed, and as you can see in the upper right corner, this is a synergy between these 2 disciplines within the Federation of Chemical Engineering, European Federation, just to work together to create some new value. And this is done by, as you can see, by modeling and system engineering applied in physiology, which can be used for describing the metabolism, the pharmacokinetics, pharmacodynamics, diagnostics, but also all transport phenomena which happen within the human body, which is very important for drug delivery systems and for therapy, and new nano and micro materials and devices for medical applications, which I mean is artificial organs, medical devices, pharmaceuticals, also point of care diagnostics, for example, using microfluidic lab on chip devices, and also other research areas that are related to development of new therapeutic strategies and pharmacological solutions. So what is important for chemical engineering, and also in this area, that we work on different scales, and we try to look at the phenomena from different perspectives, not only as engineers of the large scale production, but also from this very molecular level. And I hope that the presentation that we'll have here will show us that this is like I said. And before we start our first presentation, I just only want to mention that participants can ask questions in Q&A area, in the chat area, question and answers, by writing them. And then after the talk, I will read these questions to the presenter to make her able to respond. So this is everything from me at this moment. Also, I want to thank Martin for this preparation, all these activities, not only this one, but the activity of the section as well. Thank you, Martin. So I think that we have almost 2:10, so I can introduce you Maria Norberta de Pinho from University of Lisbon in Portugal, who will present the first talk about metabolic clearance of uremic toxins in the artificial kidney. So Maria, please share your presentation.
Yes, share. Yeah. And share. Yes. Share.
Okay.
Yeah. Are you seeing the screen? It's okay.
Everything
Okay, first of all, good afternoon. And first of all, thank you very much for the organization of these spotlight talks. I'm really very much excited about the way chemical engineering is evolving. Yes, because I think that we have a lot of challenges in chemical engineering. Okay, so I will talk about the clearance of uremic toxins in the artificial kidney, and as we, oops, I have to. Okay, so the artificial kidney, it is connected or associated to hemodialysis, and as we see in this slide, we are very far from what our kidneys performance of how they perform, because they filter a large amount of fluid, 180 liters per day, and in hemodialysis we are bound to have treatments 3 times a week or for 4 hours, and we remove in these 4 hours only 2 to 5 liters. Besides that, there are a lot of other challenges, and the main challenges are, first of all, the fact that with the conventional hemodialysis, we remove efficiently the low water-soluble uremic toxins, but the ones that they are here with the red circle, the middle-sized molecules and the protein-bound uremic toxins, they are a problem, and they are not removed, particularly the protein-bound uremic toxins. This represents solutes or toxins, small toxins that bound preferentially to the plasma proteins and the albumin namely, and then they are retained by the ultrafiltration membranes, and they accumulate in the system. So this is one of the big challenges to improve hemodialysis. And then, the other one is that we are really trying to have membranes that have higher fluxes, and we have the high flux hemodialysis that it's already present, and these the high flux hemodialysis, we have already benefits like removing some of the middle-sized molecules. Okay, and the other that it's always the other challenge, it's the hemocompatibility. In all artificial devices, we have this, if we have contact of blood or other fluids with a foreigner material, we will have problems. And so we try to have, we are working with the membranes and trying to produce membranes that are hemocompatible, compatible. But the hemocompatibility, it's a larger problem because all the equipment needs to be hemocompatible. Okay, so we are trying to have membranes that are non-hemolytic and non-thrombogenic, and that don't have or have low adhesion and activation of the platelets, and not chronic inflammatory responses, obviously. Okay, so having that in mind, we start, the first hemodialyzers, they were made, or the membranes were from regenerated cellulose, and it stays, the materials used in the hemodialyzers till 1917 were cellulosic derived materials. But one problem that happened exactly with poor hemocompatibility due to the cellulosic materials having a large percentage of OH groups that induced complement protein interaction, led to rethink about these materials, and we turn back to the cellulose acetate membrane, so, as we have the acetyl groups substituting some of the OH groups. But the cellulose acetate, besides being a an excellent film forming, it has limitations like chemical resistance and mechanical, low mechanical strength. So what we are doing, it's to try to produce mixed matrix membranes with introducing silica, and we will see how we will do that, to combine rigidity and thermal stability of the inorganic material with the flexibility and the processability of the cellulose acetate. Okay, so in that frame work, we did some literature review, and the silica in other applications, not in medical application, but in other applications, water treatment and so on, we have most of the cases, the mixed matrix membranes have the silica in the form of nanoparticles, and we don't want that. So we went ahead trying to have a homogeneous matrix, or to have the silica covalently bound to the cellulose acetate matrix, so having one phase only. Okay, so very simply, what we do with the asymmetric mixed matrix membranes, we want to remove the toxins and to keep the vital components of the blood, and we can tailor almost at as we want the surfaces morphologies of the dense layers, and also we can have active layers with minimal thickness, so leading to high permeation fluxes. So, and for that, what did we do? We coupled the phase inversion method, that it's a very empirical, very well-known method for membrane formation, with the sol gel process. And so we have the precursor of the sol gel added to the casting solutions, and then we try to induce these reactions of hydrolysis and the condensation, as we see in this slide. And then we did a series of membranes with a lot of different contents of the TEOS, that is the sol gel precursor. And the casting solutions are casting solutions of cellulose acetate, acetone, and formamide, to which we add sol gel precursor. Okay, so, and then we have by ATFT, we really have the evidence that all hybrid membranes present particularly one peak that it's very important, CEOC. So we have the silica covalently bound to the matrix, and that does not, of course, does not exist in the membrane without silica. So we are in the good way, and then we have here again the effect of increasing the silica content, and we increase the area of the peak that we want of this peak, but at a certain time we don't have benefits, and we will see what is happening. So with the higher contents of silica. Okay, so now testing these membranes and the hydraulic permeability, that it's the capacity of the membrane to let be crossed by water under pressure gradient, transmembrane pressure, and we, by the slope of the fluxes versus the transmembrane pressure, we get the hydraulic permeability in kilograms per hour, per square meter, and per bar. And as we can see, we have improvement of the hydraulic permeability for the membrane with 5% of silica and 10% of silica. Yes, and then for higher values, we have a decrease of the hydraulic permeability. So we are keeping with the 5% and 10%, and for some time also the 18%, but then we will see that the membranes with 5% of silica are the ones that we are interested on. Okay, so here we have the molecular weight cutoff of these membranes, and as you know, the molecular weight cutoff it's determined by the permeation of solutes with molecular weight increasing and we get the molecular weight cutoff of here a little increase for 5%, and for 10% a little bit more increase. But then for from 20, 30, and 20 and 40, we have a decrease of the hydraulic of the molecular weight cutoff. Okay, so and what we want, as we said, we want to remove the toxins and keep the vital components of the blood and the plasma proteins. And so we did the test with these 3 membranes with the 5, 11, and 18% of silica, and we really have that all of them let the urea go through completely, and they retain the Benyahia. It's we don't do it with human serum albumin, but it's the same. It's approximately the same thing. Okay, so now about the morphology and the SEM analysis of the dense surface and of the porous surface, that it's a porous support beneath the dense surface. And what we see here with 5%, it is that of silica, it is that these membranes display in the surface these stripes with a very regular mode. And then we went ahead to do atomic force microscopy and for these membranes, and in the atomic force microscopy we have the membrane with 5% of silica with the lower values of Z, RQ, and RA, that are parameters that pertain or that show, give evidence about the roughness of the surface. And in previous study for other membranes and for other for blood oxygenators, we have seen that the polyurethane membranes, in the polyurethane membranes, the decrease in RA correlates very well to the enhancement of hemocompatibility, and that it's published, and it's another polymer. But anyway, we go ahead with having smoother surface, the smoother as we can. Okay, and then about the stripes that we saw in SEM, here they are evident in IFM, and we have stripes with height of 50 nanometers and with spacing the 2.5 micrometers. And then now about the hemocompatibility, all the membranes were non-hemolytic with a HI parameter less than 2, and the thrombosis degree, as we can see here, for blood contact time of 15 minutes, the thrombosis degree of all membranes, mixed membranes, is lower than 30% of the positive control. And also the 2 membranes, with 5% and 11% of silica, show the lowest thrombosis degree for all blood contact times. Okay, so we are happy with that. And another parameter of hemocompatibility, it's the platelet deposition and the platelet coverage. And as we can see, these membranes, the introduction of silica reduced the platelet deposition from 6 to the 10 to minus 4 platelets per micrometer, 2.5. So we are a reduction on the number of deposited platelets, and also the coverage for these hybrid membranes is smaller compared to the CA membranes. Okay, okay. So now let's talk about another parameter of hemocompatibility, that it's the platelet activation, that it's described usually by these 5 stages of activation. And what we can see is that none of the membranes present platelets in the highest stage of activation. And the number of platelets in the second stage of activation is reduced by introduction of silica from 16% to 3% for these hybrid mixed metric hybrid membranes. So I will not go through the conclusions, because probably I am getting close to my time, but I just will reinforce here that we have membranes that fully permeate urea as a model of a toxin, and retain fully BSA. And these membranes are monophasic, so the membranes, the presence of silica, the silica is covalently bound to the cellulose acetate matrix given by these the presence of this peak. Okay, and then we saw some interesting features related to hemocompatibility. But I would like to use the last, I think that I'm within my time. So I would like to go to future work. Where are we aiming? We are aiming now to enhance the removal of the protein-bound uremic toxins, and we are doing that through the addition of metal organic frameworks, MOFs, that preferentially absorb the toxin bound to the albumin, so and get this toxin free, and the toxins will go through the membrane and be removed. Okay, so that it's work going on, and we have already some membranes already casted by, just as I said, just by adding these UEO 66 MOF to the casting solutions. And we did that to 2 membranes, a membrane more tighter and a membrane CA34 that it's more open as a porous, larger porous structure, and the molecular weight cut off higher. Okay, so and we have here some preliminary results, and we can see that the hydraulic permeability increases with the addition of the MOF for the CA22 for the tighter membrane, but not for the other membrane with larger pores. And then the molecular weight cutoff also increases for the CA22 membranes. I must say that the difference between these two, CA22 and CA34, is the casting solution. It's the ratio of the solvent, the acetone to to formamide. So the system of solvents, it's acetone formamide, and the ratio of that determines different porous structures. Okay, and so I think that I am very well behaved. And here we have some preliminary results, and we can see that all the removals are low, below 5% compared to higher values for the CA membranes. Okay, and last but not the least, I would like to say that now we are not only working with the membranes, but trying to develop a micro device and doing all the microfluidic experiments to have an artificial kidney that will eventually, it will be minimizing the blood trauma associated with the fluid dynamics of the membrane-based hemodialysis treatment, and also eventually to get a way of having a continuous removal of water and toxins. And this work is being done by Miguel Pereira da Silva, and then we have here the acknowledgements to all the people that have been through these last years collaborating with us, and I am grateful to the FCT due to the sponsoring of a project that it's finished, but on artificial organs, blood oxygenators and kidney, and our unit of research. Thank you very much. Yeah.
Thank you very much, Maria. Thank you for this interesting presentation. I look at this question and the answers window. We don't have any questions at this time from the audience, but maybe David has one. Yes, please, David, switch to microphone.
Yeah, happy to ask a question. Very interesting, Maria. Thank you very much for that. This might be a rather naive question because I'm not so familiar in this area, but I was just thinking it right at the beginning, this mixed, you seem to have you need to mix
matrix membranes,
mixed matrix thing, and it was about permeability and strength, and then you're adding silicon to it in some way, and I'm wondering whether it wasn't clear. I mean, is silica for a chemical purpose or is it for strengthening of the membrane, and whether there are, I don't know what the limits are on what sorts of materials you might use, but maybe, you know, by trying to mix the two, you might be getting the worst of both rather than the best of both. So maybe there's a completely new material, well, not completely new, it still has to be cellulosic, I suppose, but something quite new that really still has the strength, but allows the permeability that you require. I'm just wondering to what extent there are new materials, new possible materials out there that just might make things much easier.
Well, this is an area of intense research of the new materials, as you probably know. And the reasons why we went through this combination of the two methods, the phase inversion and the sol gel, it was it was exactly to have the reinforcement, not only chemical but mechanical reinforcement of the matrix of the membranes. And in fact, I didn't do any testing of the mechanical properties, but there are collaborators of mine that have done, and really we have improvement of the mechanical strength. And I didn't quite got your question, David, because you said that it could be the other way around. It will be.
Well, you might have the worst of both worlds rather than the best of both worlds, so that it will always be a compromise. Is there something inherent that is?
And why should we have the worst world or the?
You might. I mean, it just depends to me whether the strength and the permeability are.
The permeability increases.
Yeah.
And so the water removal, that it's an important for the equilibrium in the patients doing hemodialysis, an important thing to have these fluxes of water being stabilized. So I don't know what to tell you, David. I think that the way we have been progressing step by step, we are happy to have membranes that they are more permeable, that they are more resistant. As I said, I didn't measure the mechanical properties, but I had collaborators that did it, and there is improvement. And also, because there is this feature that it's very important, we have a monophasic matrix, because as you saw by the ATF, we see that the silica is covalently bound to the polymer. Yeah, so and a lot of silica added to organic materials for other applications. At least the literature shows that you have the conditions that we do the addition of or the salt gel. It's acidic conditions, because if we don't control exactly these conditions, we will have nanoparticles there. And I think that then we will have not a good membrane. Yeah.
Okay. Thanks very much. That's
I cannot answer
really
your question.
No, that's fine. I was just stripping it back a bit. Yeah. Thank you.
Okay, thank you. I mean, the very interesting topic that Maria showed is this increase in hemocompatibility, and you know, that is not only permeability is the same, but some other features are much superior to the original one. Okay, thank you. I think that is the time to thank you again for this presentation and for discussion. It's time to go to another talk, to the next talk, which will be presented by Professor Eva Maria Martin Valle from University of Salamanca, Spain. And yes, please share your screen. Switch to the presentation mode, please.
Now, can you see now?
Yeah, but can you switch to the presentation mode to the full screen?
It is now.
Oh, yeah.
I finished now,
Thank
right?
You.
Okay.
Yes.
Well, good afternoon. First, I would like to thank Tomasz, Martin, and David for the organization of these Spotlight Talks. I think this step for the European Federation of Chemical Engineering is to move forward and to show that chemical engineering is not feel static, but is dynamic. And finally, we can see how we can share different knowledge in order to improve the quality of patient life using our knowledge. I belong to the University of Salamanca, to the Chemical Engineering Department, and also I belong to the Institute of the Biomedical Research of Salamanca. Through my presentation, I will go through some main important numbers in cancer, how the hallmarks of cancer and the integration of nanotechnology is being the key point for chemical engineering to play an important role in cancer. I will present two important applications. The first one is based in modeling and how the fundamentals in chemical engineering, like transfer phenomena and reaction, is very, very important in the nanoparticle interaction on how the predicting models are reducing the experiments with animals. The second one is the application of chemical engineering in nanoparticle design using the cancer cell metabolomes as reprogramming, but mainly based on the reaction and catalysis as the key point. And finally, the conclusion of my talks. If we pay attention to the cancer number, we can see that today is the current leading cause of death worldwide. The last data published in 2020 point out that 19 million of cases are already diagnosed and still 10 million of deaths. We can see in the plot that it's amazing how in the last 14, 15 years, the prevalence on the society is being increased. This is thank you to the new development, the contribution of the research, and also the holistic approach of the cancer treatment. However, according to the incidence, we can see that still there are two main cancers that they are prevalent on the society. The first one is the female breast cancer, and the second one is the prostate. However, still the mortality is very related to these two cancers. It's quite high regarding the whole data. Making an early diagnosis is essential to prevent tumors from causing metastasis, because the metastasis is the main important point for the mortality. Well, current issues today in cancer treatment are based in diagnosis and lack of selectivity therapy, because several side effects and resistance contributes to late cancer diagnosis are very important. The first key point is the limited knowledge about cancer risk factors among the general population and delays in seeking medical attention. In addition, non-selective therapies refers to treatment. Both cancerous and healthy cells offer different significant side effects. These therapies are mainly based on cancer treatment and can generate different effects, not very positive on the life. Different efforts are being made to develop more acute treatment options. We can mention therapies based in immunotherapy, combination therapies, etc. However, over the past decade, the nanotechnology technology and the development of new polymers, materials based on nanotechnology has given main contribution on the attempts to develop new therapies. The hallmarks on cancer are set on different characteristics that those kind of cells are point out. Those hallmarks provide a framework that they can be applied for the fundamental change that occur in the tumor cells. Nanotechnology is considered as one promising for cancer research and treatment, offering different solutions to address several hallmarks. For example, nanoparticles can be engineering as therapeutic agent directly to cancer, to different cancer cell and healthy tissue. This is done by surface tailoring that can allow the attachment of different specific targeting ligands such as antibodies, and they can selectively bind to cancer cells and avoid the accumulation at the healthy tissue. Also, nanotechnology offers different strategies in order to avoid resistance in cancer cells. By encapsulating different compounds in nanoparticles, these therapeutic agents can be avoid the degradation and efflux pumps, improve the viability and avoid the resistance. But however, today, what is being applied by chemical engineers and also of other fields is energy, reprogramming the energy metabolic behavior of these cancer cells. I will go through later with one example in which I will show you how this catalytic reaction can be applied. The use of nanoparticles is based on the effect of permeability and rotation, EPR effect. This what allow is the penetration of the nanoparticles on the tumor tissue. To do that, we need nanoparticles with proper size, suitable size, and also surface that allow perfect interaction to generate an accumulation and selectivity accumulation in the tumor tissue. This will improve the drug delivery and efficiency. In addition, another key point that is used for targeting is the overexpression of specific compounds that cancer cells provide an opportunity to nanosystem that can selectively target therapeutic agent to these cells. Thus, those specific ligands will smartly respond to different components. This is the main basis that is used right now at the clinic in order to design personalized therapies. But the key point for this talk today is what chemical engineers are doing right now. What is the role of the chemical engineers in this field, the contribution of the biomedical engineering? It is true that if we pay attention different phenomena to analyze, since we have a nanoparticle on the body circulation, and after that, this nanoparticle is interact a specific way with the cell, we have many important steps that only chemical engineering can design and can analyze. If we go back through 30 years, we can find the first book on chemical engineering, transfer phenomena in biomedical engineering. He was written in the 70s in US, but Professor Lightfoot, that he was one of the contributors with Professor Berth to transfer phenomena in chemical engineering. But during the last 10 years, 20 years, we have many textbooks in which the fundamental of chemical engineering are very designed, and is a very important key point, because for a clinical application, in order to avoid it says that they are not very worth it, we need to have a good understanding of the phenomena, and with that we can extrapolate what the doctors are doing. See, to we pay attention of what happened with a nanoparticle is in the circulation system, we can see 4 main processes that we need to define in order to have the understanding. The first one is the interaction of the nanoparticle with the red and the white blood cells. This is very important. The second one is the absorption, especially considering the surrounding vessel and the tumor. The third one, very important, is the diffusion step, what happened in the tumor tissue, what is an isotropic system with different permeability, etc. And the final one, and this a key point, is the tumor cell recognition. But in order to have a good therapy, we need to have a better understanding of these 4 steps. To do that, it's important to analyze what happened at the pore level with interaction with the nanoparticles and the surrounding. We need to analyze, as we can see on the slide, first the collision of these nanoparticles with the cells, convention and retardation effects. I mean, to have knowledge and analyzing of the transport property of the system. Finally, those transport properties are being included in different continuum models. With that, we can have the effect of the tumor microenvironment and also the pharmacokinetics. Those data are very important in order to have a simulation prediction of the behavior of those nanoparticles in vivo. Those models are very important because they will reduce in many times, in many situations, the number of experiments that we have to done in vivo. To do that and to include those experiments, the formulation is they are typical chemical engineering, the starting low, the RC low, Kensington-Ketansky formulation, and it's obvious that the species conservation low. I mean, many fundamentals of chemical engineering in order to have a main understanding of nanoparticles circulating in the human body. The CFD is playing an important role. Is very helpful in order to understand the interaction. One of the most common methods is the inverse molecular finite element method, in which molecular interaction, electrostatic interaction, and mass transport interaction is included. I mean, the main equation under Eulerian approach and Lagrangian approach is taking place. Also with the force distribution, with the electrical forces, and also the velocity profile. This kind of system is given some important information regarding the first step, that is the key point, is the circulation of the nanoparticles, because these models will give some information about how the real number of nanoparticles that will reach to the tumor. That means is quite important. The second sample of chemical engineering is how the combination of nanoparticles and what happened with the reaction and catalysis inside the tumoral cell is keeping included together in order to have something interesting. If we are talking about reprogramming, we can say that the first step to point out that the cancer cell are behavior with a different reactor, like if we compare with the normal cell, it was in 1924 when Barbour pointed out that the energy generation, the glucolysis in tumor cells are different than normal cells. This was it was important, but it was about 15 years when the Memorial Sloan Kathleen Center is the most relevant research center for cancer, and a clinical application demonstrate that the way to approach to the cancer cell goes through the cancer reprogramming. If we consider this abnormal metabolic phenotype of the cancer cell, we can see that we can use 5 different approaches. Genetic alteration, that is so biochemical, this is not so good for chemical engineering. Tumor differences, different concentration of oxygen, pH, nutrients is important. Redox behavior, biosynthesis, and bioenergetics. I will focus through the redox behavior. During the last years, it has been proved that the reactive oxygen species play an important role in cancer cells behavior. If we have a normal concentration, everything is fine. But when we have a very high concentration of rocks, cancer cell experiment a behavior that is it's called ferroptosis. That means it's the death of the cell. In this case, we can think that if we can generate a high concentration of this, we can modify the redox equilibrium inside of the cancer cell. We can improve this ferroptosis effect. That means the death of the cell. Considering the manipulation of the rocks, we try to use ferroptosis as a strategy. our first thought it was to find a material able to absorb a high concentration of iron. Iron is playing an important role on the phenton equilibrium that is taken in the cancer cell. We have used polydopamine. Polydopamine is a synthetic polymer. It's based in dopamine and has remarkable biocompatibility, tunability, and we can modify the surface very easy. This material is able to absorb high amount of different metals. We can see here how are our nanoparticles. We can produce control size and monodisperse nanoparticle by a polymerization control reactor, in which we have proof that those nanoparticles can absorb high amount of different metals. But our metal is iron, which is going to modify the equilibrium of the phenton inside the cells. The hypothesis is like that. If we have the cancer cell and we have our nanoparticles will internalize. When we are at the lysosome, they will modify the phenton equilibrium, kidnapping the iron. This modify or the equilibrium will increase the ROX production and generate ferroptosis. That means the death of the cancer cells. We have been working in HER2 plus breast cancer, that is one of the most damaged cancer in women. The prevalence is only 3-5 years, and we have been working on systemic therapies and also local therapies. By today, the therapy at the clinic is the combination of antibody, trastuzumab, and also a Taxol derivative, paclitaxel. Both are very toxic inside the body, and the side effects are very complicated. Our approach was to combine the reprogrammation of the ROX, generation of ferroptosis, with the clinical therapy that is using by now. To do that, we have produced under control our nanoparticles size. After that, we tailor the surface with PETX, is the paclitaxel by chemisorption, and after that, we incorporate the monoclonal antibody. This therapy was tested in vitro, in 3D spheroids, and in vivo. We compare the behavior with the tumor cells and also with healthy cells, not only to see the efficiency, but also to see the selectivity. It's very important to check how those therapeutic agents are selective over the cancer that we are studying. We can see if we compare with the healthy cells and with the tumor cells, that the efficiency over 3 days, 72 hours, it was over 15. We only remains 15% of the cells alive. The same results we check, we compare with the spheroids, and we can see in vivo also that it was working. That means the combination, the synergy with the chemotherapy and also with the metabolic reprogramming is being a promising technique. This kind of nanoparticles were compared also with other common treatment, that is doxorubicin. In this case, we won't compare if we could modify the phenton equilibrium, the iron equilibrium, if we absorb in our particles iron. We prepare the nanoparticles in the same way with the incorporation first of the iron, and after that the chemotherapy. In this case, we did the same experiments, and we observed that the efficiency of those nanoparticles were lower than when the nanoparticles were naked. That means that the best way to modify the equilibrium is to kidnap the iron. That means that we have been many experiments, and we are studying the equilibrium in order to understand the kinetic rate of this system. Well, in conclusion, at this brief exposition, it can be stated that the contribution of chemical engineering in cancer treatment is very important. The main fundamentals of the different situation and different phenomena they are taking place on the body are very important. The human body is the most complicated chemical reaction industry, with so many reaction, transfer phenomena, absorption taking place at the same time. It's obvious that in the new years, in a few years, the contribution of different fields under this holistic approach will generate very good treatments, ensure that, and the quality of life of the patient will be very much better. Because the definition of engineering is use of technology to improve the quality of life of the society. Many thanks.
Oh, sorry, thank you very much. I couldn't switch my microphone on. Thank you very much, Eva, for very interesting talk, which exactly addressed what we have in mind when we're talking about chemical engineering and medicine. So it was very interesting, very inspiring. And I'm looking to the question and answers, but still nobody. Oh, David, he's raising his hand.
Always willing to ask a question. I mean, I've worked a little bit on these things with our Cancer Institute, but not so much. But a couple of questions. One was, if I've got time, let's just start with one. One of the things that happens, they're quite complicated metabolisms going on, and the drugs tend to target specific links in the metabolism to try and prevent cell reproduction. I think a future, from what I understand, particularly when they have, they evolve and they mutate and all these sorts of things, is that because cancer seems to be quite smart, you need to pick off a number of targets in the network. What is the limit of these nanoparticles? Can you have as many sort of therapeutics in there as you like, or is there a sort of limit to the number of, could you have 5 or 10 different therapeutic agents within the nanoparticle, which could then somehow magically target the pathway in the cell that needed to be suppressed?
Well, thank you, David. It's a nice question. Well, every day we know more about cancer. We are really working with clinical doctors. What happens now is when a patient is diagnostic of cancer, the first thing that they did is a molecular analysis. From the molecular analysis, they made a screening of different genes and different overexpression. Together, at the same time, they are running different clinical studies. That means from the information together of the molecular analysis, the gene, and different therapeutics, we got the information. And they let us know, we want to try this in order to reduce the concentration. For example, the case that I saw, this is a clinical trial, we reduce in the nanoparticle 10 times the concentration that they use at the regular doses in systemic way, what is very important because we improve the selectivity and also reduce 10 times the concentration. That means by today, we can see, for example, for HER2 plus breast cancer, we have at least 5 targets. The first one is the biggest overexpression over the membrane because it's the way to have a specific interaction. But the information, we don't have it. It's very biomedical, very biochemical. They give to us the information put together, molecular analysis of different patients, and also different clinical trials. It's very complicated. For example, this antibody trastuzumab, it was about 3 years for the company in US in San Francisco with 200 postdocs in order to develop this antibody. That means the bottleneck is the clinical trial. I mean, most of the drugs going through the 2 steps, but to go through the 4 is quite complicated. But they are making many progress. I mean, the screening, answer your question, the screening of the molecular analysis of the tumor is very, very crucial today to find a personalized therapy.
Interesting. Thanks.
Thanks.
Got a rather interesting paper, which I'll send you, which I read, which was very interesting in looking at the how they could tackle the time scale of the cell reproduction as well. I'll send it to you because it was extraordinary, and the most huge amount of work must have gone into that one paper, I think. Anyway, I thought that was an interesting, from a dynamics point of view, that we could be that would be an interesting problem for chemical engineers as well.
Yeah, it's so complicated. The modeling is so complicated because you have to reduce the numbers of phenomena that is taking place. I mean, but what I want to say is modeling is quite important. I mean, it's not giving you the exact response, but it's giving you an overview of what is going on inside. And this is very important in order to reduce the experiments with animal models.
Classic systems engineering, isn't it?
Yes,
we can't do everything. We can't model everything. You have to decide what is the minimum subset that is going to do something useful. Difficult, but
very difficult, but making improvements. Thanks.
Okay, thank you. I liked what Eva said, that the human body is the most complicated chemical industry.
It's the most complicated and the most clever.
Okay.
Thank you.
Well, thank you very much again. And I think that we can go to the next presentation, which will be given by Dr. Roberto Abbiati from Boehringer, Ingelheim, from Germany. It's quantitative systems pharmacology. So, please, Roberto, share your screen.
Good afternoon, everyone. I trust you can see my screen.
Yes.
Yeah, thank you. Thank you for everyone in the audience today, and a special thank you to the organizer of this section of chemical engineering as applied to medicine. I think, as it was iterated today, this is a very good time to see chemical engineering application in this field, and I think there will be a large impact in the next future. I think also there is quite a variety of the application option that we have as chemical engineering, and I think this is very much highlighted in the presentation today. So today I'll stress one of these potential applications, which is formally called quantitative system pharmacology. This is a component of pharmaceutical sciences, which is strongly based on modeling. And today I would like to give a brief introduction to this discipline and also stress what's the relation between this discipline and chemical engineering and which are the contact points. So before we look at specific application, I'd like to set a little bit the stage for this presentation and to clarify which is the context of application of this discipline. So we are focusing on drug development, not so much on manufacturing or production as one may think as chemical engineering. That means in most cases, we have a drug molecule that has been identified and we want to make it suitable for clinical application. It has to be a compound that is safe and it has to be efficacious. And this activity has been done for many years in the past. So one may argue what's so relevant at this point in time and why quantitative system pharmacology could be so relevant here. One of the factors is the change in the type of pharmaceutical products that are becoming more and more utilized lately. These are very peculiar properties, both in terms of pharmacokinetics, that means evolution of concentration level of this drug in the body, and also pharmacodynamics in terms of how these drugs are efficacious. One example could be, for instance, CAR T. These are chimeric antigen receptor T cells. These are basically leukocytes that are modulated in order to target a specific target in the body. For instance, in most cases, is cancer therapies. Or for instance, nanoparticles, these are vectors which are loaded with drugs and have a very high affinity for a specific target, or viral vectors and so on. We can really bring a lot of examples that may be put in contrast with standard, small molecules that have maybe a more restricted range of features that we need to characterize. Here are safety and efficacy, right? We want our drug to be safe for the patients. We really want to determine which are the toxicity limit in which we want to administer this drug, and also what's the range of efficacy, relate the efficacy to the dosage of the drug. And obviously, these are two important factors which are also constraints for the clinical studies, right? Once we want to develop a compound, we need to conduct a series of clinical studies, and we have limitation on how much we can test this drug, both in animals and then experiment in humans. And then, of course, quantitative system pharmacology can support in that direction as well. Also, there is an increasing amount of data becoming available. We have a large amount of omics data. We have very nice imaging tools that provide us with a lot of data, but quite diverse data. And then how can we optimally combine this data? And we want anything we do, we want to do it fast. We want to bring it rapidly to the patients to try to really tackle the ongoing health issues. And this brings me to the definition of quantitative system pharmacology. So this is a relatively young discipline. I think we could argue that the start could be traced back 2011. There was a NIH white paper that was kind of setting the stage for the application of quantitative system pharmacology in the pharmaceutical industry. In terms of definition, quantitative system pharmacology is a mechanism-based approach to translational medicine that integrates experimental and computational methods to elucidate pharmacological concepts. And this is it's quite complex definition. I would just like to stress a few concepts. So it's a mechanism-based approach in translational medicine. So it's trying to convey knowledge that has been collected preclinically through different set of experiments and make it applicable at the clinical level by integrating both experimental and computational methods. So there is a computational methods. This is maybe the primary link to chemical engineering. There is a strong component of modeling, but there is also a strong integration with experiment and availability of data. Now, if we want to look at the founding concept of quantitative system pharmacology, we typically define mathematical models that try to describe the biology and the physiology of a given system. It could be an organ, it could be a disease state, and we really want to reproduce the full complexity of this organ or this disease by capturing the individual components. And this is why this is often referred as a bottom-up approach. That means we start by individual components, and we try really to identify what's the relation between components. This could be, if we refer to a biological system, we could have cytokines, we could have cell types, and these interact with each others. And by bringing this together into a mathematical model, we want to be able to describe the holistic behavior. We have component of translation, as I mentioned before. This is allowing us to link preclinical to clinical application. And we want to identify different sources of knowledge, as I mentioned before, large variety of very different data that are becoming available. On the right side, we just have schematics. This is, just descriptive, but what I would like to stress here is the multi-scale concept that we have in systems pharmacology. With the fact that we really want to link the individual components, we also have this multi-scale concept, which is also inherent to chemical engineering. So once we describe, for instance, an organ, we also want to describe all the levels of interaction from molecular stage to cell to different tissue, going all the way up to, individual patients and maybe patients' populations, which are very important for personalized medicine. This structure can be modeled and integrated with experimental data. The combination of the two is what we typically refer as QSP, or quantitative system pharmacology. This is already a self-standing type of product, but we can combine it with a specific drug for which we have, as I mentioned before, PK and PD, pharmacokinetic and pharmacodynamic information, and generate prediction and projection of what could be the behavior of the system or the behavior of a treatment. So now the question is what we apply this for. We can apply this to create a mechanistic rationale to understand the disease onset, the disease progression. We can also explore alternative mechanisms of action, right? If we know how the disease is represented, is progressing, we can also try to virtually modulate this disease, targeting different mechanisms of disease progression and see how that would play out on the long term. We can also combine this with specific drug information, pharmacokinetic, pharmacodynamic, try to predict the efficacy, toxicity, or maybe optimal dosing and schedules. There is another factor here that is key, is the variability of an outcome. Every patient is different. The disease progression across patients is different. We want to be able to capture that to identify patient subpopulation that will benefit the most from a specific treatment. And this is something we can also do with quantitative system pharmacology. And of course, the idea is that we want to provide rationale to support decision-making, make a drug to the patient faster, and reduce the risk associated to the development of a compound. Now, you may be wondering at this point how the quantitative system pharmacology model looks like. And these models are quite diverse. Here I'm trying to summarize one of the main features, some of the main features, and also try to link it back to chemical engineering. So most of the QSP models are based on system of differential equation, most typically ordinary differential equations. These are based on compartmental structures. On the right side, we see an example. I won't go into much detail here. This is an example taken from literature. This is a large ordinary differential equation-based model of non-alcoholic fatty liver disease. It's really capturing all the steps of onset of the disease, progression of the disease, and also the component of the immune systems that are involved in inflammation and fibrosis. Here you see, multiple boxes. Each box is a specific component of the disease. It could be, for instance, a cell type. It could be neutrophils or macrophages. And the model represents the interaction between these cells and the disease progression component. Now, in case you model this with this approach, this is based on ordinary differential equations, which from a chemical engineering standpoint, it resembles what we know as a continuous stirred tank reactor, CSTR. There are obviously, this is the model structure, then there are functional relationship across component of these schematics, which are again based on native concepts that are intrinsic in chemical engineering, like the law of mass action, reaction kinetics. If you think about catalyzed reactions, there is also a component of catalyst, which we obviously can represent with absorption, surface absorption, and so on. So we have a number of standard equation forms that go into the definition of these models. Then we want to parameterize the model. We do that by leveraging different type of data sets. And then we can constitute what is commonly referred to as virtual patients. Virtual patients is a definition that we often apply in quantitative system pharmacology. A virtual patient is a specific parameterization of a model that represents a specific disease status. So if you generate multiple parameterization of the model, with different disease onset, different disease progression rate, you can in principle represent different virtual patients and then test your treatment in different patient populations. So this is a very common application. It's quite relevant for personalized medicine. There are also two major ways to apply system pharmacology models. Something is referred as a platform model. These are models which are very broad in scope. They represent the full complexity of a disease and can be applied for, strategic decision where you want to target different potential pathway of disease progression. if you want maybe to impact regulatory application, or you want to have a very specific analysis, typically we go for a fit for purpose approach, where we take a subsection of our very complex model, we qualify the model for a specific application, and then we apply it to generate prediction that can support maybe the filing of a drug to a regulatory agency. Now, what is the status of QSP in pharmaceutical industry? I mentioned this is a relatively young discipline, but still was able to make quite a bit of impact, both in academia and also in industry. So most of large pharmaceutical companies have a QSP group. Maybe mid-sized pharmaceutical company may have a small QSP group internally, or maybe outsourcing the activity to quite a number of CRO, consulting research organizations, that are available for this type of activities. It's a highly interdisciplinary activity because there is a modeling component that requires certain type of skill set, but there is a large amount of information that need to come from different interfaces, from strategy development, therapeutic area, and medical advice. We may have biologists and experimentalists that would generate data and provide expert input into the model. So it's important not just to develop the model, but to be able to interact with different interfaces. Input data may come from experiment, it may come from literature. And again, what do we eventually apply it for? It could be for internal testing and exploration, to support internal development. It could be to support a regulatory submission. And there is also, guidelines that are generated by regulatory agency, like FDA, for instance, that try to define which is the range of application of quantitative system pharmacology, and try to outline a little bit the requirement for its applications. This is still, to try to link it back to chemical engineering, or more specifically process system engineering. On the right side, we have a typical image that is always nice to look at when we make this link. This is actually an old work of art that represents the human body as an industrial plant, and this is really, the idea that is going into the definition of many of the quantitative system pharmacology models. So these models, as we said, they are multi-scale. They can be represented as a connected set of reactors with different characteristics. We have mass transport phenomena. We have processes of receptor binding, competitive bindings, and we certainly have a component of fluid dynamics as well. For what concerns numerical aspects, we deal with large system of differential equations. This system may be stiff. We need to have appropriate software tools and skills to utilize these models. Very many times we cannot fully parameterize the model. We need to undergo through a process of parameter regression. We need to know how to utilize sensitivity analysis, global optimization. So there are different levels of skill set that are necessary for a scientist in this field. Now, I like to bring a couple of examples from industry to be a little bit more concrete here on the application. This is a publication from the pharmaceutical company Merck, the American pharmaceutical company, where they were demonstrating the impact of modeling into their pipeline. And they brought a number of examples. Here, I would just briefly like to stress a couple of them, or maybe specifically this one related to the assessment of the risk of bone fracture. So they had, they were in a trial and they wanted to be able to assess what's the probability that a bone in a patient will undergo fracture, because the drug that they were developing, odanacatinib, was supposed to reduce the probability of fracture. However, how do you assess that? That really takes several years for you to observe a fracture in a patient. What you can do, however, is to collect image of the bone structure of a patient, characterize it, and maybe utilize modeling tools to predict the probability of a fracture. And that's the idea they came up with. They utilize imaging tools to determine the structure of the bone. They reconstructed the bone structure with finite element analysis. So in this case, we are dealing with partial differential equation. And with almost a mechanical engineering approach, they could do virtual tests of the probability of a fracture of the bone under a given load. This allowed them to predict the probability of the bone fracture. This is what it's called a surrogate endpoint. It's very important, for clinical development. And what they did was to file by proposing this as an exploratory surrogate endpoint. This is a very nice application with a very high level impact in the clinical drug development. Now, they also listed what's the impact of modeling and simulation drug development. You can support dose and schedule decision for different phase, primary, phase 1, and phase 2. You can reduce the time for the development of the clinical trials, and also you can provide the rationale to justify the dose selection. You can support by equivalence, so deciding whether a new generic drug has equivalent products characteristic to a commercialized product. You can also look at some toxicity aspects like TQT. So these are all enabling decision. You can also apply modeling to support no-go decision, right? The development of the drug takes a long time. They cost hundreds of millions of euros. It's also important if you can determine upfront when a drug may be a little chances to make it to commercialization, and therefore decide to invest your resources in another direction. Here is a second example that I would like to bring you on quantitative system pharmacology. And again, this is an application that requires both partial and ordinary differential equation, and is primarily focused on what we call pharmacokinetic. In this case, we were trying to predict the distribution of a drug-loaded nanoparticle into solid tumor sites. And of course, this is important because one of the factors in targeting solid tumors is the difficulty to bring the drug to the actual tumor site. In order to tackle this problem, very often we utilize nanoparticles that have very specific characteristics to target this tumor. But then the question is, how can we predict the time and the penetration of this nanoparticle into the tumor? So what we can do is to try to reconstruct the full process of drug transport to the target site. Here we have on the bottom left the schematics. I will briefly describe what is showing here. So we have a drug that is loaded on a nanoparticle. What typically happens is when you put a nanoparticle into the blood is that blood proteins will absorb on the surface on your nanoparticle, and the original native characteristic will be modified. There may be a release of the cargo. The cargo will be your actual drug. And this is already a problem by itself in the peripheral circulation. Then your nanoparticle will travel through the blood vessels to ideally the site where the tumor is present. There will be a process of extravasation. That the drug will go out of the blood flow into the extracellular matrix in presence ideally of tissue cells and also of the tumor. At that point, we have a process of interaction of the nanoparticle with the tumor, potential internalization and release of cargo. So there are many different aspects to this model. Some are drug specific, like the material of the drug, the size, the surface charge. Some other parameters are specific to the tissue or the tumor itself. So the model allows us to account for both parameters that we can control, typically drug specific parameters, and parameters we just have to account for, like typically tissue specific parameters. We can come out with the model with different level of complexity. In this case, we had an approach where we had a finite element model representing a simplified tumor structure surrounded by a layer of normal tissue. We are seeing a cross-section of a spherical geometry. We could parameterize the different region of the tumor depending on what we know are characteristic, like the level of heterogeneity, the blood vasculature, the interstitial pressure of the tissue, then we can come out with a set of equations. In this case, it's a combination of mass balance equation with Darcy law for fluid flow driven by a pressure gradient. We can also have the Starling equation for transport of species across a membrane, in this case, the vessel microvasculature. So of course, this is also published and is referring in the bottom of the slide. And this allows us to predict in time and space the concentration of the drug inside the tumor in its different forms, as a drug loaded into a nanoparticle or a free standing drug, and therefore what would be the concentration of the drug over time. We can also compare this prediction to clinical data. Of course, typically in this case, you would measure the total concentration of the drug in a tumor, whereas the model can provide drug-specific information, both in time and space. And this is another example of how system pharmacology can support pharmacokinetic aspects of drugs here. Now, I'm moving a little bit to draw the conclusion here. And these are some considerations for chemical engineering, that would like to apply their skill set into translational medicine. So as chemical engineers, we typically have an approach that allows us for rationalization and being also pragmatic in the model development. We have skill set in designing, the mathematical model and implementing that. We can also typically break down the system into the individual components, but keeping in mind, the global dynamics that it is derivation of the individual components. And of course, we have knowledge of physics and chemistry and technical skill of numerical analysis. But when we go in the application of translational medicine, we also need to pick up concept of biology and pharmacology. It's also important to have the understanding of what's happening in the laboratory, where the data are generated, which are the limitation there, because that's important for us to understand the data that we want to incorporate into the model. We need to understand there is a level of uncertainty and variability that we typically don't see when we model chemical plants. And we also need to think a bit in probabilistic terms. There are so many different outcomes. There is so much variability across patients. And we also need to be able to communicate with a quite diverse audience. Now, closing with some perspectives, QSP is still young, but it's growing fast. It's being appreciated both in industry, in academia, and by regulatory agency. For chemical engineers to make an impact there, some specific education is needed. The classical chemical engineering skills are very important, but it's important to appreciate the value and the importance of aspect of biology and physiology to really be able to critically develop a QSP model. There is also some work to do, formalize methodology, workflow for developing QSP models. It's important, model reproducibility. There have been publications in the past showing that models that have been published and for which code was available in the literature were actually not reproducible. This has been a topic and still is a topic that the community is working on. It's important also to establish a repository for sharing knowledge and allowing colleagues to utilize models that have been previously developed, because after all, physiology and biology are unique. Also develop new methods to integrate different type of data. There is a large amount of omics data becoming available. It's also important to leverage new techniques like machine learning into classic ordinary differential equation-based models, and also define better the interaction with regulatory agency and their requirement. And with this, I'm closing. These are just some acknowledgements to colleagues for which I've been interacting with in the past. And I really like to thank you for your attention, and I'm available for any question you may have.
Thank you, Roberto. It was a very excellent talk showing us this new field, new discipline that is evolving, that is growing, and you showed also the strengthness of chemical engineering as a discipline which brings tools to let's say deal with these problems that you showed us. Okay, I look at the question and answers. Still nobody is wants to ask.
I can ask a question.
David
I can always ask a question, unless Eva wants to ask a question. She might, because she's a bit closer to this. But that was particularly, I mean, thank you so much. That was really interesting. It was really good to see so much going on here. Just to talk to the audience as well, we just need more. It's quite complicated, as Roberto was putting across, and so we need more people trained in this area, chemical engineers going through doing master's level and PhD level qualifications. I think to really get on top of all of this, so my question, it's quite a sort of general question. I was wondering where you think the big challenges are for chemical engineering research that might help you take a step forward. Where do you see the biggest sort of barriers? You mentioned about omics data and data sharing and stuff like that. Yes, obviously, and machine learning, those are important as well. But I was thinking, and we're pretty good at drawing those things, but I'm thinking of what are the more core chemical engineering things that you think are really not able to address the problems so far as you see them?
Sure, thank you. That's a really good question. So one of the limitations that we are typically facing here is that these are models that have mechanistic in nature. They can grow quite complicated, and we need a very diverse and detailed amount of data to characterize this model. As a matter of fact, many times these data are not available because they need to be sampled in a living body, either preclinically or clinically. That's not always possible. Sometimes we don't have the tools. Coming from chemical engineering, maybe it's a situation that is not so commonly faced, because in a chemical plant, first of all, that was a human design in the first place. Second of all, you can most often really, with a good level of accuracy, determine most of the parameters of the system. So I think really defining a pipeline to support the design of experiment, maybe in a model-based fashion, to generate the data that are suitable for the model and to optimally integrate that. We mentioned the aspect of omics data and machine learning. These are certainly tools that are supportive, but I think there is still room to generate data and to measure data in the laboratory, integrate them into the model. But it's a long-term activity, and these are data that needs to be planned upfront before the model starts getting developed. So it's also let's say it's a conceptual approach to this discipline. We need to interact with other colleagues in the laboratory, support them in the design of the experiment, and generate the right data point, the right piece of data that will go into the model. So I think this could be a factor that would help the discipline itself, but also that the chemical engineering approach to this discipline has to account for.
Thanks.
Yeah, thank you.
Okay, thank you. Thank you. So I think that we will now go to the last talk in our webinar. And I will ask Professor Brahim Benyahia from Loughborough University, UK, to present his talk right now. Yeah, the title is on the screen. Okay, go ahead. Brahim, your microphone is off.
So sorry, I was just saying I'm very grateful to you and to David and the whole group for this opportunity, hoping this will reach a much broader community. I'm also very grateful to the previous colleague who led the ground to my presentation, particularly Roberto, who he really raised some of the issues I'm going to talk about. So in essence, I'm going to introduce or motivate. Sorry for that. So I will motivate or introduce my work and try to convince you that there is a lot of room for chemical engineering to help us develop much faster, much more effective pharmaceuticals. And I will specifically talk about a model-based DOE framework that we have developed within our group and validated experimentally. And then once we have the model, of course, the best thing to do is to design the process and optimally operate the process. And that's what I would like to talk about. And I will I will talk about some of the issues, particularly in the space of formulation and crystallization, such as shape control and modification. And this is again another framework that we have developed. And with this, we can also think about the optimal design of the whole process or integrated processes. And I will wrap up with some concluding remarks. So before I start, I would like just to highlight some of the key facts associated with pharma. As mentioned earlier, it's really costly to bring a new drug into the market. It takes very long time, usually between 8 to 12 years. It costs more than 2 billion. Currently, we are talking about $2.5 billion. And clinical trials are really very challenging. It used to be based or focused so much on blockbuster, and we thought that the era of blockbusters has ended, but now it's getting back, particularly post-COVID. And particularly what is also important in pharma, it's really stringent in terms of regulation. We have to pay attention really too much to the drug safety and efficacy. Another key fact is most of the drugs available now are solid dosage forms. So it depends on countries, but usually we think about more than 60% of the drug products are oral, solid oral dosage forms. So this highlights the importance of formulation and the importance of crystallization, which is one of the main purification processes used in pharma. Unfortunately, pharma is still dominated by batch, despite of all the advantages that has been shown through continuous manufacturing. So there are a lot of good signs now. Pharma is embracing continuous manufacturing, but we are still at the beginning. Again, pharma is heavy in terms of experimentation. They use loads and loads of experiments, and we know that APIs or intermediates, reagents in pharma are pharma grade, and they are extremely expensive. And we would like to address some of these problems. Not to mention that pharma is also solvent intensive, and it's probably the least environmentally friendly industry currently. So again, as I mentioned, crystallization appears to be the most important purification step. And I will convince you that it's not only a purification, it's one of the most important formulation steps. And again, drug efficacy strongly depends on the production process and formulation. That's why we need some of the toolkits available in chemical engineering to address some of the key challenges that I have highlighted. So in terms again of crystallization, you may expect crystallization to happen in many different places. We believe that more than 80% of drug product involve at least one crystallization step during their manufacturing. So if we pick up any process, we may expect crystallization followed by filtration and drying. And we have also the drying, the downstream processing, where these particles produced during the upstream will be used in terms of formulation through the granulation, whether it's wet or dry granulation. So the powder obviously will impact the quality of the tablets, and it will directly impact drug safety and efficacy. So it's really important to get the particle size and particle properties within specification. And when we talk about this specification, we are talking about crystal size and size distribution. We are talking about crystal shape and shape distribution, purity particularly, and thus quite common. Most of the reaction in pharma, they generate side products, and they may even generate neurotoxic impurities that are really very harmful, that may even lead into failures during clinical trials. In addition, we need also to get the right polymorphic form, which will determine as well the solubility or the dissolution rates, and it will determine the shelf life or stability of the product. In addition, all of these will impact manufacturability. We know that plate-like or needle-like crystals, they do not flow quite well, and it's really hard to filter these particles. So we need to get them, as close to a sphere as possible. We are talking usually about aspect ratio. We need to get the aspect ratio as small as possible. Most importantly, all these properties, which are referred as critical quality, attributes of the crystallization, the impact, the final critical quality attribute of the drug product, such as uniformity, friability, disintegration, dissolution, and of course, drug safety and efficacy and stability and shelf life. So we need to get these within or under control. And particularly when we face the problem of plate-like and needle-like, which are quite common in pharma, we may use different techniques such as co-crystallization, spherical agglomeration, the use of additives or solvent modification, temperature cycling, milling. And as you see, there is a lot of decision-making here, and sometimes it costs a lot of money to try these different techniques. So we need to develop at early stage, particularly during clinical trials, we need to get the production process right in order to avoid any delays in the launch of the product. So when it comes to talk about chemical engineering, I'm sure people, what comes to people's mind is usually mathematical modeling, process design, optimization, scale up, minimization of cost, minimization of the development, and so forth. So I will not spend a lot of time talking about this, but I'm pretty sure that chemical engineering will play a key role in terms of bringing Industry 4 into different industries, particularly pharma. But what I would like to highlight today is particularly the use of optimal design of experiment, particularly model-based design of experiment, which will lead, once we have more reliable and predictive models, we can develop more optimal designs and more optimal operations of the process, which will also open up opportunities for better control of quality and better control of the whole plant. So crystallization is an important step, as I mentioned, and it's considered as the very early formulation stage, because this is where we determine the particle size, distribution, and all other properties I have mentioned earlier. However, crystallization is very challenging because you have lots of phenomena taking place at the same time. This may include primary nucleation, secondary nucleation, growth, agglomeration, breakage, and so forth. So in an ideal world, we need to have a perfect control over all of these, but at least we need to have predictable models that capture the knowledge or capture the underlying phenomena, which will help us design more effective processes. And that's the main purpose of one of the slides I'm going to present today. So one of the things we have developed in our group is a model-based design of experiment. And usually, when we start, we have usually a set of possible models. So we have a dilemma. We don't know which model would be the best model, and we need to determine which model we're going to go for. So first of all, we can implement what we call structural identifiability. Without spending a lot of time talking about the math, the whole idea here is looking at what can be measured, what are the outputs that we can measure. This will inform whether the parameters of the model can be identified basically based only on the structure of the model without using any data at this stage. So this approach will help us to do two things. The first is to discriminate between models, and it will also tell us whether the analytics we are using are enough to capture the knowledge that we need. So once we do that, we can implement a test. If the parameters are not structurally identifiable, we can loop back our feedback and determine or identify another model or pick up another model. Then we may use some initial experiments that can be also available in the literature, but if we are developing a new drug, that means we have to run some preliminary experiments, which will help us develop or determine the initial parameters or parameter estimates to be more precise. So with this, as I mentioned, you can have different loops, and these parameters will become the nominal parameters based on which we can develop a model-based design of experiment. So this model-based design of experiment, which I will explain in a few seconds, will use the information of the sensitivities, basically Fisher information matrix, and it will help us explicitly minimize the uncertainty in the model, which will guarantee model prediction capabilities at the end of the day. So this will help us determine the optimal experimental campaign that we need to run. That means the minimum number of experiments at lower cost, but experiments that contain maximum information content, and that's what we need. So with this, we can determine the new parameters, the parameters that are more representative of the mathematical model, and then we can run a different test. If we are happy, we can move forward. Otherwise, we can loop back either to the initial step, pick up a different model, or we redesign the experiments. And to do that, we can also use an estimability analysis, and this is what we have implemented in our group. We have developed a multi-objective optimization that is based on the minimization of Fisher matrix, information matrix designs, or I will explain this in a few seconds. But at the same time, we can also assess whether the information in the experiments is enough to identify the parameters, which is called also practical identifiability. So if it is satisfactory, then we stop. Otherwise, we can loop back again and collect new data. Right. So there are many different techniques that can be implemented that are based on models to determine the best experimental campaign. This may include the A-optimal, E-optimal, and D-optimal designs of experiments. All of these, they are based on the minimization of the confidence domains. In other words, it minimizes the space within this elliptical uncertainty or confidence domains. Of course, there are better ways to represent uncertainty, but this is based on some approximations. The elliptical ones are reliable enough. In our case, what we have used is the D-optimal design of experiments, which essentially minimizes the determinant of the inverse of the Fisher information matrix. And again, this is what happens once we run it. The whole idea is to minimize. But of course, minimizing the experiments may also come with a cost. We need also to pay attention to correlation, and this is why implementing estimability in particular will help us reduce correlation. So in short, implementing model-based design of experiment will help us minimize the variance and maximize the information content within the experimentation. To validate the approach, we selected a batch crystallization process. Here we are using paracetamol in water. And of course, we are using a complex mathematical model that involves many different kinetics, growth, agglomeration, and so forth, which overall contains 14 parameters. So the whole idea is to determine the best way to get the best parameters, but at the same time, minimize the uncertainties within these models. We came up with a really new design of trajectories and experimentation for crystallization, and this is currently submitted for publication. So we may consider, for instance, linear cooling, which is the traditional approach in crystallization, but we may also consider different types of piecewise linear with continuity, and most importantly, we opted for a temperature cycling technique, which at the end of the day will allow us to run one single experiment rich of data that can build the best mathematical model. In addition, we can also implement temperature cycling with holds, which will help us also capture some induction times and get the best experimentation possible. So this is the mathematical formulation of the problem. Again, I will not spend time talking about this, but I'm certainly happy to give more or shed more light on this if you have any question. So in essence, this is what happens. By implementing the D-optimal design of experiment, we can guarantee smaller minimum uncertainties and smaller confidence domains, but at the same time, we can minimize correlation between parameters. I'd like just to highlight the fact that higher correlation within your model leads generally into a very poor prediction capability of the model, because what happens is some parameters may compensate the effect of other parameters, and that means the model is not sensitive to the simulation or to the data you are feeding into the model. So this is what happens with the validation, and the pink dots here represent the sampling points suggested by the D-optimal design, and these are validated. So these are the experimental points versus the model predictions. And as you see, temperature cycling is the input, and all the others are the output, the concentration, the total crystal count, the number of particles, and the mean crystal size follow quite nicely the trajectories that are shown by the experiments. Now that we have a good or reliable mathematical model, the next step is to use a design. And again, design is one of the fundamentals of chemical engineering. And one of the approaches we are using is the attainable region. In essence, the question we are trying to answer is how many continuous crystallization stages we may need, particularly in the context of moving from batch to continuous, and what are the individual and total volumes for each stage, what are the flow rates, particularly for anti-solvent crystallization, and operating temperatures. So this is a very complex problem, and we would like to know, before we invest, before we buy very expensive equipments, we would like to know what is the range of capacity and how many stages we need. And if you look at here on the right-hand side, it shows us 2 stages and 3 stages, and shows the attainable particle size, which will tell us what can be achieved at the end of the day if we pick up one design or the other. So once we are happy with this, we have validated the approach by looking at a more complex system where we consider 5 stages instead of 3, but we have also allowed a very interesting thing. We allow dissolution, which mimics what we call temperature cycling in crystallization. And we looked at minimization and maximization of the particle size, because in industry, sometimes the objective is to maximize the particle size, but sometimes it can be the minimization, particularly for powders, for inhalation and so on. So as you see here, the optimizer tells us exactly what are the temperatures that we need to set up for the different stages and the different volumes. Once we are happy with the design, the next step is to optimally operate the process. And one of the challenges encountered in chemical engineering in general is the startup shutdown of optimization. And we wanted to implement some of these techniques in pharma. So here we looked at 3-stage or a 3-stage continuous crystallizer. We also considered seeding. That means we have particles that we seed into the first stage, taking into account different seed particle size, different seed concentration or density, and so forth. So the number of decision variables is huge in this particular case. And we looked at different possibilities in a systematic way, how to start up the process to minimize the time to reach steady state. This is really important because in pharma, it is expected that the operating window will be very small. People talk about 30 days, for instance. So compared to oil, the oil industry, where we talk about year, or 1 year or more, so the startup will be really significant, not to mention the cost of the reagents we are using in this particular case. So looking at the different scenarios here, we have roughly 18 scenarios. We managed to minimize the startup time to 14 minutes, which at the end of the day shows an improvement of above 70% of the startup time. We did a similar thing for the shutdown, and on top of that, we looked at a multi-objective optimization to see what are the best compromises for a shutdown. Here, the shutdown consists in collecting as much crystals as possible and avoid, wasting very valuable material at the end of the process. And here again, we would like to keep our particle size within specification in order to achieve the right quality imposed by the regulator. Once we finished with this, we looked at a gray transition problem where I have heard my colleagues talking about personalized medicines, and this fits perfectly well with such a situation, or with the situation of clinical trials, where the idea is to produce different products with different doses or different properties without stopping the process. And that's what we have achieved here. By optimizing the gray transition and at the same time optimizing the startup and shutdown, we managed to reduce the transition by 70%, and we reduced the startup time by 80%. So in terms of benefits, this is huge in terms of cost minimization. Once we validated this, the next problem we wanted to address is one of the most common problems, is needle-like crystals. Here we have developed a framework, so a systematic workflow to test different strategies to control and get the particle aspect ratio within the right range. So this may include different techniques such as simple linear cooling, wet milling, the addition of additives, and temperature cycling. And this framework contains all possible combinations in a systematic way. The whole idea is to escalate only when something doesn't work. And this is really nice because it avoids the industry to go through a very difficult approach when a very simple approach is enough. And here we show that the best approach that seems to be really novel here, the combination of wet milling, DNC, direct nucleation control, or temperature cycling, and additives in a certain way delivers the best crystal aspect ratio, which is really promising, compared to the initial particle size. If you look at the needle shape here, we are talking about an aspect ratio of above 50. We brought it down to 5. Now, what happens if these techniques do not work? People usually tend to avoid spherical agglomeration. We use it just as a final result. And here we have designed a process and a new way to achieve spherical agglomeration, which again can help reduce the number of steps involved in a manufacturing, leading into a much faster formulation of the final product. Here we have used for the first time a dispersion cell, because one of the biggest challenges in spherical agglomeration is to control the bridging liquid that we use in general to bring the crystals together. The best way to achieve a better control of the droplet size is to use a membrane, and this is what we have achieved. This is very important because the particle or the droplet size determines the mechanism by which spherical agglomeration happens. If we have very large droplets, usually the most dominating mechanism is the immersion, leading to large spherical agglomerates. Whereas if we have very small or tiny droplets of the bridging liquid, we tend to have the distribution mechanism, leading to a very poor distribution of the spherical agglomerates. And this is typically, in terms of design of experiment, looking at the different impact of the flow rate of the bridging liquid, the mixing, and so forth. And these are the SEM images showing very nice spherical agglomerates. We have also identified regimes under which we can produce actually hollow spherical agglomerates that may perhaps fit to certain applications. So another aspect we looked at, again, using some of the chemical engineering tools, is superstructure optimization. The whole question here is, we would like to design in a systematic way an integrated process that integrates reaction and crystallization by implementing different recycles in a systematic way. And this is typically what we call superstructure optimization. And at the end of the day, this determines how many steps we need and what are the different recycle streams we need. And we implemented a multi-objective optimization capturing yield, coefficient of variation, and particle size distribution, followed by a decision-making process to be more precise, a multi-criteria decision aiding process to help identify the best compromises. With this, we looked at, again, the big picture. The whole idea here is to produce the final tablet, and looking at all challenges associated with the design and the optimal operation of the process. And here, for instance, you have the reaction, the crystallization, followed by the granulation, the wet granulation, drying, and the final tablet press. And one of the things that we are really keen to address in our research is to bring a new concept of quality by digital design, because currently the current paradigm in terms of quality control is heavily based on quality by design, which is heavy in terms of experimentation. We would like to bring these model-based approaches and develop more reliable quality by digital designs. And this needs usually more reliable sensitivity analysis, particularly global sensitivity analysis, to determine the design space where we need to operate the process in a robust and efficient way. With this, I would like to wrap up. So in terms of formulation, and I gave you just a flavor of the formulation. The formulation of pharmaceuticals is a mix of science, engineering, and art. This is good news. That means chemical engineering may play a key role in developing the next generation formulations. The proposed digital chemical engineering toolkit that I have shown today may help reduce the cost of experimentation, as well as process and product design, but most importantly, it can enhance product quality, consistency, and process stability, resilience, and robustness, particularly when we have a market that is fluctuating quite significantly, or for clinical trials. With this, I'd like to thank my research team, particularly Jomin, Wei, Timothy, Jashu, and Isabella. Thank you very much.
Thank you very much for this presentation. I think that we have a question from Roberto first.
Yeah, I would have a question for Brahim. Thank you for the really nice presentation. And I think, this is extremely relevant for application in the system pharmacology space. I think in particular, these two aspects of model identifiability, which is sometimes overlooked, but it's highly important, both structure and practical. Sometimes we look at the practical, we look at the uncertainty of the parameter that we have estimated, and we forget that upfront we should have be more careful maybe in the design of the model structure in the first place. And the other aspect is the design of experiment. Can we support our colleagues with a model-based approach and maybe determine the time point that would allow for the better estimation of our parameters? So do you see a possibility to translate this toolkit to system pharmacology application and maybe make a tool that is available and can be easily applied? Because sometimes I have used tools in the past for identifiability. They were not always applied to all type of models, or they were difficult to interpret. So, anything that could be provided in that direction would be very useful. Any comment you may have?
I totally agree with you. I do believe it can be applied to any kind of model. You have mentioned models based on ODE, ordinary differential equations, or partial differential equations. It can help. It can be applied easily to this particular problem. As long as you have a system where you can measure certain outputs and you have certain inputs and you have the dynamics of the problem, this can be applied. So it can be applied even to CFD problems or surrogate models, a reduced version of a CFD model. Yeah. So I have shown here the crystallization, but we have done also within the formulation space with the lubrication of tablets and granulation. And it works really well. It shows that we can really reduce the experimentation burden. Yeah.
Thank you.
Okay, thank you. I will take a look to question and answers. Nobody from the audience wants to ask any specific question. So, David, you don't want to ask the question. You asked 3 questions before, so okay. Well, let me thank you all of the speakers of today, and let me try to summarize what we had here. I'm very grateful to all of you that you take an effort to show us your research, your data, and I'm happy that we covered different fields of this chemical engineering applications to medicine, from specific medical devices, let's say, or developing of the smart treatment of cancer, up to this quantitative system pharmacology, which was also very interesting, up to optimization of pharmaceutical design and industry, let's say. So this is really a very broad scope, and I'm happy that it was like that we could see all these aspects. Of course, there are not all that we want to cover in this section, but they are very from different perspectives, from different scales. And what is important, and it's of course what's showed by many of you, that first of all we need to learn a lot from medical doctors. We will need learn a lot from biology, biochemistry, maybe much more than typical chemical engineers should know. And then we use our background from chemical engineering, modeling, describing the equilibria, the kinetics, mass transfer phenomena, and so on. And then we also need to learn how to communicate with other people from other disciplines, because this is very interdisciplinary. So many of you showed that, so I'm only summarizing. And well, I think that is a very good event to make a progress within our section. And it was said in the beginning, many of us will meet in Berlin, so we can talk about the future of this section. And David, maybe I can ask you to summarize it better than me.
No. I think you've done it very well. Thank you all to everybody. I wanted to add one thing, that the section hopes to have a meeting next year as well. I'm not sure exactly when. I think we were talking about in the spring. Probably we were offered to go to Salerno in Italy. It's a very strong group there. So I hope we'll see you there. Anyway, it's really great to see such activity and really seeing chemical engineering really starting to get into medicine. And of course, as Tomasz says, our interactions and our collaborations are going to be very important, because we certainly can't solve all these problems, but working with our clinical and non-clinical colleagues in medicine is going to be really important. So with that, we'll just wind the meeting up, I think. And thank you all again very much for your contributions, and thanks to the audience, and thank you, of course, Martin. And we will see you hopefully see you all in Berlin.
Thank you. Bye-bye.
Thank you. Goodbye, everyone.